Thank you very much. Thank you, CGC, for this opportunity. So I'm going to present on behalf of the Human Functional Genomics Project. I'm going to present today on the genetic regulation of the inflammatory proteins, explaining on the different disparities that we have seen between the Tanzanian and the Dutch populations. I have no disclosures. So this is just an overview of how the Human Functional Genomics Project, specifically from the Tanzanian cohort. So it was a recruit among the healthy individuals. So there will be among the healthy individuals, so it was around a cohort including of 350 study volunteers and participants. Then later on, we ended up just with sufficient volunteers who agreed to participate, and then blood samples were taken, the inflammatory proteins was measured by the all-link inflammatory panel, and protein expression was measured, and then further PQTL mapping was done, followed by overlap and meta-analysis in comparison between the two cohorts. Now, QC was done on these two different populations. So the Dutch cohort had a slightly higher number of 500 participants, but then a few participants were lost along the way. So we ended up with along 340 samples in the Dutch cohort and 286 in the Tanzanian cohort that met the inclusion criteria and had genetic and inflammatory protein data from both populations. So we say there is inter-individual variability even within the same population. That's what we see. But we wanted now to see maybe perhaps there is more than just lifestyle or behavior, that the genetic factors have an influence on this. So from previous studies, what we also had seen, there is a difference in the way the inflammatory proteomes were expressed among the East African and the Western European cohorts, where we see there is higher inflammatory protein expression in the Tanzanian cohort in comparison with the Dutch cohort, where we were able to observe some of the inflammatory proteins, such as 4EBP1 and FG21, have slightly higher, and where you can see the Dutch cohort, we have a slightly lower inflammatory protein expressions between these two cohorts. So now the question was, why do we see this? Is there maybe a reason that is more than just a different lifestyle, maybe the different dietary that happened between these two cohorts? So we started just wanted to see if, to assess the heritability between these two populations, to see the extent of the genetic variation that is being contributed. We wanted to assess now the proportion of the observations that the variations that we see in the inflammatory proteins that is actually attributed to the genetic factors. So we started by estimating the genetic relationship matrix, and with heritability being expressed as 0 and 1. So this is just a plot showing the SNP heritability between the two populations. So the top one is the Tanzanian cohort, and the second one is the Dutch cohort. So right from this, on the x-axis, we have the inflammatory proteins, where we see there is already variability between this one. But what do we say from this? Is it like really true that there is like 99% or heritability, that the environment or other pathogen exposures have no effect on this? So it got us alarming, but then one of the reasons for this may be probably due to the small number of sample size that we had from these two cohorts. Then we thought that it's best that we further explore this and see now we do quantitative locali mapping between these populations to see to identify maybe the specific genetic variants that are associated with the variations on the expressions or the abundance of the proteins that we see. Now, if it was just we wanted to focus, identify the specific genetic regulators that have a slightly significant influence on the protein expression. So, from the Tanzanian cohort, we were able to identify 10 loci that were genome-wide significant, but also from the Dutch cohort, we were able to identify also 10 loci that were slightly significant. So you can see from the Manhattan plot that we see there is slight differences. We thought maybe perhaps this is also caused by the slight differences. You have about 100 fewer samples between the Tanzanian and the Dutch cohort between these populations. Maybe that's why we see. But despite of these ones, the genome-wide significant SNPs that we saw from these both two cohorts were completely different. So it got us to think, okay, does that mean that there is actually no overlap between these populations? Taking into account that both of these populations were healthy populations, they were screened from the HIV negative, normal blood sugar levels, normal pressure, etc. So we found out that just at a 5% level, there was the overlap between these two populations. It was not so much. It just ranged from 0 to less than 2%, so 0 to 1.5% overlap between this among all the 72 inflammatory proteins that they were similar between these two cohorts. Then if we wanted to be more like stringent and just at the suggestive level to see if there are some specific inflammatory proteins that we were able to see and observe if there is any overlap in the loci between these two populations. So at the suggestive level, it was streamlined a bit, and we just ended up with 6 of the inflammatory proteins, with IL-18R1 being has the most loci that are shared between this cohort, about 26% on that. But then we still see CCL25, which also had the most loci at the 5% level. So we still see, even at the stringent suggestive level, we were able to still see the overlap between these two cohorts. Then we further confirmed with a colocalization analysis of the inflammatory proteins between these two cohorts. We confirmed 9 inflammatory proteins have any shared loci between these. So among all the 72, it's only 9 that have shared. So we used the different cutoff points that you can use for colocalization, but for this we use the one that's a posterior probability that is at least greater than 0.75. So we are able to identify again CCL25, CD6, CST5, CD8A, CCL3, and IL-18R1 again. So we thought that these are the inflammatory proteins that we keep seeing that, okay, so these ones, its regulation have any impact on genetic regulation between both of these two cohorts. So we see these are the only ones that have shared. So it's still not much. It's small between these two cohorts that we see. Then we further wanted to identify the loci that show any effect on plasma proteins in the same allelic direction. Now, in both of these populations, what we did, we actually meta-analyzed the summary statistics of each of these markers across the two cohorts. Now we wanted from the two cohorts After meta-analyzing, use the mixed effects model. Only the variants with a p-value of 1 times 10 power negative 5 were included. From the meta-analysis, we identify these 10 genome-wide SNPs that have that were genome-wide, but we also identified several other that are suggestive from this. So from these results, what we also see again, we see IL-18, RR1, but also CCL25. So based on these results, we thought that as part of the larger group from the Human Functional Genomics Project, but for this case, we wanted to analyze the data from the Dutch and the Tanzanian cohort among the healthy population. We saw that there is a clearly distinct difference in the individual variability of the inflammatory proteins that were expressed. But this is also one of the first studies to be done to be able to identify the genetic inflammatory protein factors among the East African population. So we were also able to identify some novel or perhaps ancestry specific variants that we could not find in the previously published databases. So this brings into light the importance of including underrepresented populations, especially in genomic studies. So this is a cohort among the healthy population, but it also serves as a baseline that can also be used for other further research studies within that. I'd like to thank all the participants from the Human Functional Genomics Project, the amazing Human Functional Genomics team, our UMC and KCMC partners. Thank you.